And welcome to this lesson on enzymes, a very complicated type of molecule with a simple job. Enzymes make important chemical reactions in living things happen more quickly. For context, let's look at where we've been. Where have we seen enzymes before? We've seen them in the protein pathway. Recall that we have our nucleus that's sending out instructions to build proteins. Ribosomes have been building those in the endoplasmic reticulum, and then those proteins get transported out, reconfigured, and sent throughout the cell. One of those special types of proteins is our enzymes, which we have pictured here. Now, the shape of an enzyme can be very, very complex, but we're going to simplify it. Let's have a look. On the left, you see our cartoon version, and on the right, you see our complex 3D model of what one of these enzymes might look like. Very simply, our enzymes are going to have some sort of active site, a location on that enzyme where another molecule or multiple molecules can come in and either be put together or be broken apart. Those molecules that are coming in we call the substrate, and perhaps they're coming in as two parts that join in that active site and then leave broken apart as products. Either way, the enzyme is acting on the substrate, either to put pieces together or take pieces apart. So just note that enzymes have that active site and different stuff's going to come in and get changed. So here's your definitions. Our enzymes are specialized biomolecules, usually proteins in this case, there are some exceptions, acting as catalysts in biochemical reactions. They make chemical reactions happen at the cellular level. Now, we just introduced another word there, catalyst. What's going on with that? Well, a catalyst is any substance that speeds up the rate of a chemical reaction. And here's the key part, without being consumed by the reaction. You can reuse enzymes, a type of catalyst, many, usually millions of times. Now again, I've said this a few times, biology is not a chemistry course, but little elements of chemistry come into what we study. Let's take a quick look at what's happening with chemical reactions just in general, the idea of products and reactants. A chemical reaction, when we say this, we're saying that atoms of one or more substances are being rearranged, resulting in one or more different substances. We're going to read our reactions from left to right. Reactants are the items on the left, and the products are the items on the right. You may have one or more reactants, you may have one or more products. In this case, we have some sort of hydrogen and oxygen and carbon coming together to make this new carbon, hydrogen, oxygen compound. Let's have a look at our example. This is showing you a cycle of an enzyme acting on those things that we just saw in the chemical reaction. In this case, H2O and CO2 are our reactants. And they're making something new. We have this product over here. What's happening in this reaction? Well, those two reactants are coming in. Those are our substrates. They are binding to this enzyme. And that little opening there, we call its active site. Now, the active site is actually bringing those molecules close enough together to rearrange them, rearrange the bonds in those different atoms of those molecules to make something new. Here you see the same materials, looks like 3 oxygen, 2 hydrogen, and 1 carbon to make this new product, the carbonic acid. This enzyme, so carbonic anhydrase or anhydrase, can take water and carbon dioxide and make carbonic acid. And because this is a catalyst, once it's done its job, it can go grab another couple molecules and go through this again and again and again and again. Enzymes are catalysts that make little chemical reactions happen in living systems. Now, one thing to know about these catalysts is they're making a reaction that's already spontaneous, something that would happen more quickly, or at least at a rate that's what should be happening in a cell. We call this concept activation energy and a reduction in activation energy. Let's have a look. Activation energy, if we graph it out, is the energy we need to start a chemical reaction. So in this case, in this graph, we have reactants starting off at a high potential energy and ending with the products at a low potential energy. If this had just been its own hill, you could imagine that if there was a little car or something approaching, it could just roll downhill. This is a reaction that would happen on its own. However, reactants turning into products, usually there's some sort of energy cost involved. We call that the activation energy. We have to get uphill, so to speak. Catalysts are going to help us with that little section there having to go uphill. Here you go. The activation energy is what we need to reduce. Catalysts are going to help us with that. This image shows you here a comparison of what is needed for activation energy with or without an enzyme. In the blue, we've got our reaction pathway without the enzyme. The needed energy requirement is much higher. But with the enzyme, we have a lower activation energy, less of a hill to push over. And in each case, the reactant and product potential energy is the same. No real difference here between the start and the end. It's just the catalyst helps us take an easier path. So that's what enzymes do, acting as catalysts. Let's look at how we can change these in biological systems. You have another activity that shows you a bit more with this, but I've got a couple graphs that help to drive this home for us. Changes in conditions such as temperature and pH can greatly affect the activity of enzymes. You've got a couple different graphs here, and one of them is showing enzyme activity in relation to temperature. I've got my enzyme in orange and my enzyme in blue. This one enzyme here apparently has its peak activity at a fairly low temperature range. Between the temperatures of, say, maybe that's 6 degrees and 24 degrees, it can do its job. It can work. The enzyme will act as a catalyst. It can keep reactions going. But there's kind of a sweet spot where it's going to do its best work. Somewhere there between maybe 10 and 20 degrees, so maybe around 15 degrees Celsius, that's the optimal temperature for that enzyme. This blue enzyme here also has an optimal temperature, maybe somewhere around 35 degrees. There's a temperature where it works well. Now, this is interesting to know in biological systems because it relates to how your body works and how other bacteria and their enzymes work. You may be familiar with storing your food in a refrigerator. Enzymatic activity in bacteria gets slowed and stopped at cold temperatures. The type of bacteria that could cause harm to you, their optimal temperature is usually closer to body temperature. So if we can store our food in a refrigerator, the type of bacteria that could harm us generally can't grow, live, or thrive at those low temperatures. You also see this a little bit when you're running a fever. If you run a high fever, it actually interrupts the enzymatic activity of bacteria that may be causing an infection in your body. Raising that body temperature knocks them a little bit off of their A game, knocks them off of optimal when you're feeling sick. One other item here, and maybe not as familiar to you, is pH. pH is a measure of acidity. You're very acidic, stuff has a low pH, and your low is and your high pH is more basic. If something is more acidic, it's going to have a lower pH. Here you have another couple of enzymes where they've got optimal pH ranges. This item here, maybe this is a digestive enzyme. Your stomach, you may have heard of stomach acid, also has enzymes in it that help to break down your food. Pepsin, for example, takes a fairly low pH to work. If you ever have any kind of issue like acid reflux or any other kind of digestive issue where your stomach acid is affected, it can change the way that other enzymes, digestive juices, if you will, are able to break down food. This one here seems to have a optimal range of around a pH of 2, whereas this one over here seems to function best around 6.5. That would be a very dilute acid there, almost neutral. Different parts of your body are going to have different pH ranges. And if you don't have optimal pH, the enzymes that are supposed to work there, they're not going to be able to do their job. So there's two big ideas with the enzymes. Their catalytic activity is going to be related to optimal conditions. Are they running at the right temperature? Are they experiencing the correct pH? Now, why does that matter? Well, let's go back to the shape of our enzymes. Shape matters. If a protein gets out of shape, or in this case, a specialized one like an enzyme, we call that denaturation. And the text is a little small there, so let me write it again. Denaturation is just a fancy word for we lost our shape. In the picture, you see an example of denaturation happening with the protein in somebody's hair. Somebody tried to bleach the hair blonde here, and the pH of this bleach or this process of trying to take the color out of the hair was just too much for the protein in that hair. So in some cases it starts to break or melt or become brittle. It's just not good there. This person's going to have a lot of hair breakage. They're not going to like the results of what this treatment was because they were a little too harsh on the protein in their hair and they caused denaturation. Now, I don't want to confuse you here. Hair is not a catalyst. Hair is not enzymes. Enzymes are just one type of protein. And this is a good way of showing you what can happen to a protein when those pH environments or temperature is adversely affected. Now, I showed you here someone getting their hair treated with, I guess, some sort of bleaching process here. So there's a pH change happening. Acidity was off. This can also happen with heat. If you heat treat your hair too often or with too much heat, you can cause very similar damage. Denaturation of the proteins. All right, we're almost done. Let's sum it up on the last slide. Enzyme functions. Enzymes are here to accelerate reactions that help us build molecules or to help us take apart molecules. They can do either. Later on, you may hear that those are metabolic versus catabolic processes. They help build or take things apart faster. An enzyme's structure and its composition allow it to fit together with the substrate molecules it affects. If a protein gets denatured, if it loses its shape, it's not going to be able to connect with its substrates. If it loses its shape because of excess heat or too much acid, it's not going to react very well. That's it for this lesson. I'll see you in the next one.